tomorrow?
Yes, sir. This is part 3 of the Human Anatomy and Physiology final exam review. Let's keep fighting the good fight. Keep working hard. Let's do this.
Describe the major functions of muscle tissue. Describe the structure, location in the body, and function of skeletal, cardiac, and smooth muscle.
So with skeletal muscle, we have a structure of multinucleated structure where there are striations, and the location they're found in our skeletal muscle, contributing to voluntary movement. And our cardiac muscle is only location in the body is on the heart, and this has striations on it, just one nucleus, and it's got those intercalated discs to form that functional syncytium in order to allow for the cardiac muscle to contract as one, as a full unit. And then we have our smooth muscle, where there's no striations in there. We can find these on the walls of hollow organs. So the digestive tract is a huge area for smooth muscle. You can also find it on the erector pili. And instead of Z discs, it has dense bodies to anchor the actin and the myosin.
Describe the organization of skeletal muscle from a cell to whole muscle.
So with our skeletal muscle here, we have our epimysium, right? The epimysium is going to be the outermost layer of the skeletal muscle, and it's going to wrap a bundle of these fascicles that you see right here. So the epimysium wraps a bundle of fascicles, and it contains dense, irregular connective tissue. The paramysium is what wraps a single fascicle, and it wraps a bundle of these muscle fibers here. All right, and then paramysium has dense, irregular connective tissue to it. The endomysium is what wraps each individual muscle fiber, and you find it in between each of these fiber. And the endomysium wraps a bundle of these myofibrils, these individual little dots that you see right here. The myofibrils have the sarcolemma as their cell membrane, and the cell membrane is what is going to synapse with a motor neuron to undergo the neuromuscular junction. And the endomysium is made up of fine areolar connective tissue. So as you can see right here, this is kind of a little bit of a zoom up. You have your sarcolemma. The endomysium is all around this as well, but the sarcolemma is on the inside of it, and the sarcolemma is what wraps each one of these myofibrils, and that's what the myofibrils are going to synapse with. And that's what's going to synapse with a motor neuron to undergo the neuromuscular junction.
Describe the components within a skeletal muscle and describe the structure of the neuromuscular junction.
All right, so this is the anatomy of a muscle cell, right? We have our sarcolemma, our cell membrane, where we're going to find voltage-gated sodium channels and voltage-gated potassium channels. We got our presynaptic neuron, where the action potential is going to come down and open up these voltage-gated calcium channels. We have our vesicles, which are going to contain acetylcholine that are going to release into the synaptic cleft and bind to ligand-gated sodium-potassium channels on the motor end plate. Then I go over here, we have our voltage-gated sodium, voltage-gated potassium channels. We have our T-tubule, where the action potential will travel down and then hit the sarcoplasmic reticulum, where calcium is going to be released back up the T-tubule to bind to troponin. So we have our sarcoplasmic reticulum, which is an extension of the smooth endoplasmic reticulum of cells. This is just a special type of endoplasmic reticulum because it contains calcium ions inside of it. And a T-tubule combined with the sarcoplasmic reticulum is called a terminal cisternae. And then moving on over here, once the calcium goes back up, this is what actin looks like. We have our actin, we have our myosin binding sites, aka active binding sites. We have our tropomyosin, which is covering those myosin binding sites. And we have our troponin, which is on the tropomyosin. And then we know that when calcium binds to the troponin, the tropomyosin peels off of the myosin binding sites, leaving room for myosin heads to bind to those myosin binding sites. All right, so here we go. Here's the anatomy of a sarcomere. A sarcomere goes from Z disc to Z disc, okay? And anchoring off of the Z discs, you have thin actin filaments, and anchoring off of the M line, you have thick myosin filaments. And then you have your titin here, which contributes to elasticity of the muscle. Your H zone is from actin to actin. It's important to note that in I bands, you'll only find thin actin filaments, but in the A bands, you'll find both thick and thin filaments. When the muscles are contracting, the myosin heads pull the actin closer to the M line, which shortens the I band. You think of the maze runner, the gait shortening. That's exactly what's going on with the I band. The I band shortens, but the A band does not shorten.
What is the neuromuscular junction? Well, in order for these 4 individuals to do this, and these 2 individuals to be able to do this, 23, FRN has recovered over 6,000 pounds. A whole lot of things are going on molecularly to make that possible. We are going to dive into what exactly makes these things possible. So to start out with, we have an action potential that comes down the neuron that is connected to the sarcomere. And this action potential opens up voltage-gated calcium channels on the axon terminals. Now, a voltage-gated channel is going to open whenever there is a change in membrane potential, which is another way of saying an action potential is there, and that's what's causing a voltage-gated channel to open. In this case, it is a voltage-gated calcium channel that's going to open up on the axon terminal, and it's going to cause all of these calcium ions to come into a presynaptic terminal. So all these calcium ions come on in, and these calcium ions are going to bond with vesicles. Now, inside of these vesicles contains the neurotransmitter called acetylcholine. Now, acetylcholine, once the calcium binds with the vesicles and interacts with them, the vesicles are going to fuse to the presynaptic membrane on the axon terminal, and the vesicles are going to release neurotransmitter acetylcholine via exocytosis into the synaptic cleft. Once all the acetylcholine makes its way into the synaptic cleft and comes out of the vesicles via exocytosis, the acetylcholine is going to bind to ligand-gated sodium-potassium channels. Now, a ligand-gated channel in general is a channel that is going to open when a chemical or a neurotransmitter binds to it. In this case, we have acetylcholine that is binding to the ligand-gated sodium-potassium channel, causing it to open up. Before we talk about sodium and potassium ions moving through these channels, we first have to make sure that we understand what a sodium-potassium pump is. Now, the way that the sodium-potassium pump works, to put it briefly, is that it establishes resting membrane potential. The way that the sodium-potassium works is that it moves 3 sodiums into the extracellular space, and it moves 2 potassiums into the intracellular space. This creates the resting membrane potential that we could see here on this graph, which is usually around negative 70 millivolts. So if we understand how the sodium-potassium pump works and how it establishes resting membrane potential, that is going to make the process of these ion movements make more sense. So before any ions move, the outside of the muscle cell is more positively charged, and the inside of the muscle cell is more negatively charged because of the work by the sodium-potassium pump that establish resting membrane potential. Now, once acetylcholine binds to these ligand-gated channels and they open up, sodium is going to influx the cell. It's going to influx and it's going to come in and it's going to keep coming in and it's going to keep coming in. And guess what? It's going to keep coming in. At the same time, potassium ions are going to leave the cell and go into the extracellular space. However, the important thing to note is that sodium influx is going to be greater than potassium efflux. That is what creates the graded potential, which is called the end plate potential in this graph. The graded potential, or the end plate potential, is built up by sodium influx at the motor end plate via the ligand-gated sodium-potassium channels. It's getting positive because more positive ions are going inside of the cell, which is depolarizing it and building it up to where threshold is. The buildup to threshold is called the end plate potential. So this process is going to keep happening. Sodium's going to keep coming in, Potassium will keep leaving. And then once the membrane has depolarized enough to where we hit threshold, that is when a wave of depolarization is going to occur across the sarcolemma, and it is going to open up voltage-gated sodium channels. The opening of the voltage-gated sodium channels happens because we've hit threshold, which releases an action potential. The opening of these channels allows for further sodium depolarization of the sarcolemma, further depolarizing, getting more and more positive. This is happening across all voltage-gated sodium channels, which are these purple channels in this picture, in this video. And as that's happening, we are in this phase of the graph where threshold has been reached, voltage-gated sodium channels have been opened on the sarcolemma, allowing for further depolarization and sodium influx. Once we've hit threshold, an enzyme called acetylcholinesterase is going to come in and kill all of these acetylcholines in the synaptic cleft and remove the acetylcholines that are opening the ligand-gated channels on the motor end plate. So as further depolarization is occurring because of the sodium influx, voltage-gated potassium channels are also going to open, and potassium is going to efflux from the sarcolemma, and it's going to go out. And this process is going to repolarize the membrane, and we're going to get more negative again. Eventually, our sodium-potassium pumps will reestablish resting membrane potential. So when threshold is hit, the action potential is going to be sent all throughout the sarcolemma, and it's going to travel down, and it's going to hit what is called the T-tubule. And the T-tubule is an invagination of the sarcolemma, where it allows for action potentials to go deep into the muscle cell and hit what is called the sarcoplasmic reticulum. The sarcoplasmic reticulum is an extension of smooth endoplasmic reticulum in muscle cells. It is a special kind of endoplasmic reticulum because it stores calcium ions inside of it. Once the action potential hits these sarcoplasmic reticulum, do the same thing on the other side. It's going to travel down, go down the T-tubule, hit the SR. Once that happens, calcium is going to leave the sarcoplasmic reticulum, and it's going to come over, and before it binds, we got to understand the anatomy of actin. So before any calcium binds to anything, we got to understand that actin molecules have these things called active sites, and active sites before calcium bondage are covered by the protein tropomyosin. The tropomyosin prevents myosin heads from attaching to actin and prevents anything from contracting. We have our calcium ions that are going to bind to troponin, and then once calcium binds to troponin, it is now going to look like this, where the tropomyosin has slid off of the active sites on actin. The active sites are free for myosin heads to bind to them. Once we have this setup, we can now go into excitation-contraction coupling. With excitation-contraction coupling, we start out with cross-bridge formation. We have an energized myosin head with ADP and phosphate on it, and the myosin head is binded to the active sites on actin. Then for the next stage is called the power stroke, and for that power stroke to occur, the ADP and the phosphate release from the myosin head. When you slingshot a rubber band and you let go, it releases energy and the rubber band goes forward. That is what is going on with this. ADP and phosphate, once released, pulls the myosin head forward. It does its power stroke and it pulls the actin closer to the M line on a sarcomere. The next stage, and in order for the myosin head to detach from the actin, we need ATP. ATP is going to come in, it's going to attach the myosin head, forming a cross-bridge detachment. And then ATP is going to hydrolyze and is going to recock the myosin head back to its original position. ATP hydrolysis just means you put a water molecule on this ATP and you split it into ADP and phosphate. That process re-energizes the myosin head to then be able to attach to actin. And then the process can repeat itself again. So that is the neuromuscular junction and excitation-contraction coupling. Explain the factors that are believed to contribute to skeletal muscle fatigue. Compare and contrast the metabolism of skeletal muscle with that of cardiac and smooth muscle. Compare and contrast the anatomical and metabolic characteristics of type 1, type 2A, and type 2B fibers. Define the following terms: tension, contraction, twitch, motor units, and myogram. Interpret a myogram of a twitch contraction with respect to the duration of the latent, contraction, and relaxation periods, and describe the events that occur in each period.
All right, so this is our myogram, okay? The myogram just talks about the latent contraction and the relaxation periods of muscle contraction. So during the latent period, we have no tension because at this point in the NMJ, the calcium has been released from the SR. It's going back up the T tubules. It's on its way to bind to troponin, but it is not yet bound to troponin, right? So that we have no contraction tension as of yet in the latent period. But in the contraction period, this is when the calcium is bonded to troponin, pulling the tropomyosin off of the myosin binding sites / active binding sites on actin, and the myosin heads are now binding to the actin. So it's binding to the actin and it's pulling the actin closer to the midline, which is why we have an increase in contractile tension. And then we get to the relaxation period where we obviously decrease our contraction tension because the calcium is coming off of troponin and it's being actively pumped back into the sarcoplasmic reticulum. It's active transport using ATP, just like our sodium potassium pumps, because in the SR, there's already a high concentration of calcium, and it needs to be pumped actively because the calcium's going against its concentration gradient to get pumped back into the SR.
Interpret a myogram of tension versus stimulus frequency and explain the physiological basis for the phenomena of trepe, summation, and tetanus.
All right, so this here is called a trepe, right? So a trepe is basically, you can see the increase in tension here with this graph. A trepe is when contractions become stronger at the same frequency of action potential firing, okay? So action potentials are going to be fired all along here at the same frequency with the same amount of time apart. But contractions are going to get stronger and stronger because even at the same frequency of action potential firing, the calcium's not going to have enough time to get pumped back into the SR. So more calcium gets released, and more calcium is already in the sarcoplasm to bind to troponin, which is why at the same frequency we can increase contractile strength. So that's what a trepe is. Now let's talk about temporal summation. So in temporal summation, you can see individual twitches right here. So this is tension versus time, and we have our action potential firing here and here, causing that increase in tension. Now, temporal summation is basically just increasing the frequency of action potentials occurring closer together, okay? So instead of an action potential occurring here, an action potential can occur right before this goes into full rest. So instead of here, the action potential can occur right here, and it can build the tension. And same thing can happen before that one goes to rest, and that just builds the tension. Same thing before that goes to full rest, and just builds the tension. So that's temporal summation. An incomplete / unfused tetanus looks like this. We have time versus tension, rather tension versus time, and we have something that looks like this. Okay. This is when the action potential frequency is occurring very close together, and that's why we have such a huge increase in tension with not that much time of rest. Now, a complete tetanus or a fused tetanus is when the action potentials, so the graph will look like this. The action potential frequency is going to be so close together that you cannot distinguish one action potential from another. It's like if I wave my finger really fast like this. You can't distinguish where my finger is at every single millisecond that I wave my finger really fast like that. Same concept that's going on with action potentials in a fused tetanus. You can't tell how much time has passed, or you can't distinguish one action potential from another because they're occurring so close together, which is why we have no rest in a fused tetanus. It's all, you're rising up to the maximum amount of tension that your muscles can produce, and there's not enough time for rest.
Interpret a myogram or graph of tension versus stimulus intensity and explain the physiological basis for the phenomenon of recruitment. All right, so the phenomenon of recruitment. So when we have a very strong stimulus, like it says in number 10 over here, that means that we're going to recruit more and more and more motor units, which if we recruit more motor units, they're going to synapse, more muscle fibers are going to be activated and then synapsed with. And because of that, we're going to create a higher contraction, okay? And the less stronger the stimulus is, where we recruit fewer motor units, which means fewer muscle fibers are going to be activated and stimulated, meaning that we're not going to get as high of a muscle, we're not going to get as much tension in our muscle contraction. And this graph over here just shows how small motor fiber units are recruited first, and they recruit, there's not as much tension produced, but with the medium ones, when they're recruited, they recruit they're recruited second and they produce more tension. And the large ones, like the ones in our quads, for example, they recruit last, but they produce very high tensions. Interpret a graph of the length-tension relationship and describe the anatomical basis for that relationship. Compare and contrast isotonic and isometric contraction. Compare and contrast concentric and eccentric contraction. Describe the sources of calcium in smooth muscle contraction and explain how an increase in cytoplasmic calcium initiates contraction, and compare the signals that initiate smooth muscle contraction to the signal that initiates skeletal muscle contraction. So before we get into smooth muscle contraction, it's important to understand the anatomy of the smooth muscle. So here's our smooth muscle. We have these folds that are called caveoli, and these caveoli are where voltage-gated calcium channels are going to be located. And this is where, when an action potential comes down, it opens up these voltage-gated calcium channels for calcium to come in and continue from there. And then you also have the sarcoplasmic reticulum that is not as developed as in skeletal muscle, but when the action potential hits the smooth muscle, calcium comes out of the SR, as well as through extracellular space through these channels. You also have these things called dense bodies, and these dense bodies replace basically the Z discs in skeletal muscle. This is where the intermediate filaments are anchored off of, and it is where the actin and the myosin are anchored off of in our smooth muscle. So this is smooth muscle contraction, okay? In smooth muscle contraction, we're going to have an action potential, just like in skeletal muscle, come down and stimulate the smooth muscle. So this is going to open up the voltage-gated calcium channels. It's going to open these guys up. And then extracellular calcium is going to rush into the smooth muscle, and calcium in the SR is going to also come out of the smooth muscle. And this calcium is going to activate calmodulin, right? So we're going to go from an inactivated calmodulin to activated calmodulin. And then this activated calmodulin is going to activate a kinase, turning it from an inactivated kinase to an activated kinase. And that activated kinase is called myosin light chain kinase, otherwise known as MLCK. And then this MLCK is going to phosphorylate myosin heads. So we're going to take ATP, and through our MLCK, we turn that ATP into ADP, and we take the phosphate that comes off the ATP and we put it on myosin heads. So it's going to look like, here's our myosin heads, and we're going to have a phosphate on there, and then our actin is going to be right here. And then same as in skeletal muscle, we will do our cross bridging to contract the smooth muscle. Now, to relax the smooth muscle, we basically take MLCP, and the MLCP pulls the phosphate off of the myosin head, so it dephosphorylates the myosin head. The MLCK it turns back into an inactivated kinase. Activated calmodulin turns into inactivated calmodulin, and calcium is then pumped back into the SR, and it's pumped actively out of the smooth muscle back into the extracellular space. And we use its active transport. We use ATP because when we're going into the SR, when we're going to the extracellular space, we're going into an area where there's already a high amount of calcium, meaning we're going up/against our concentration gradients, meaning that we're going to need ATP to do active transport to get these calcium ions back to where they started from. Explain how the muscular system contributes to thermoregulation. The biggest thing to think about for this LO is freezing, right? So let's say it's 0 degrees, super, super cold out. You are going to shiver, right? And the reason why you shiver is because skeletal muscle movement generates heat. Okay, so the movement of the skeletal muscle, the action of shivering, is what generates heat to make you warm and to increase your core body temperature.